Bismillahir Rahmanir Rahim. Psoriasis and obesity are two different but interconnected conditions that share complex pathophysiological mechanisms, often creating a vicious cycle that worsens each other's effects. Understanding the complex relationship between these conditions is essential for effective management and treatment. Welcome again to Derma Immunology. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University, and our video today will be about obesity and psoriasis. Here is my main reference in this video, update on obesity in psoriasis patients that was published in Life Basal October 2023. Psoriasis is a chronic inflammatory skin condition affected by genetic, epigenetic, environmental, and lifestyle factors. It is marked by increased epidermal turnover leading to thick erythematous scaly patches. Obesity is a metabolic disorder characterized by excessive adipose tissue accumulation, with its pathophysiology involving a complex interplay of genetic, environmental, and hormonal factors. Recent research has demonstrated a connection between psoriasis and obesity, with each condition potentially triggering the development of the other one in a bidirectional relationship. Several adipokines secreted by adipose tissue are elevated in both obese and psoriatic patients, showing mechanisms of action similar to those involved in the pathogenesis of psoriasis. Also, specific human microbiome patterns have been identified for both obesity and psoriasis, potentially representing future therapeutic targets for selected individuals. Excess body weight also can impact both the treatment response and the occurrence of adverse events in psoriasis, which in turn may reduce patient compliance. The immune response in psoriasis is marked by the proliferation of T helper 1, T helper 17, and T helper 22 cells, leading to the production of pro-inflammatory mediators such as interferon gamma, tumor necrosis factor alpha, interleukin 6, interleukin 17, and interleukin 22. Due to the systemic inflammatory status in psoriasis patients, several comorbidities are associated, including cardiovascular diseases, metabolic syndrome, obesity, diabetes mellitus, and psoriatic arthritis. Obesity is diagnosed by calculating the body mass index, which is the weight in kilograms divided by the square of the height in meters. According to the World Health Organization, body mass index between 18.5 and 24.9 is considered normal. A body mass index of 25 to 29 indicates overweight, and a body mass index of over 30 denotes obesity. New evidence suggests that the obese phenotype should not be determined only by body mass index, but also by the individual's metabolic state. A metabolically unhealthy normal weight phenotype has been recognized and is at risk for meta-inflammation. White adipose tissue secretes various adipokines involved in metabolism, appetite regulation, inflammation, and immunity. The most studied adipokines in psoriasis are adiponectin, leptin, and resistin. Due to the pro-inflammatory nature of adipokines, except for the anti-inflammatory adiponectin, obesity is considered a low-grade inflammatory disease, which forms the basis for several comorbidities. Overweight and especially obesity are recognized as independent risk factors for developing psoriasis. On the other side, psoriasis itself can also contribute to the onset of obesity. Here is some important shared pathophysiologic factors between psoriasis and obesity. Number one, inflammation. Obesity triggers low-grade chronic inflammation, which can exacerbate psoriasis by promoting cytokine release and immune system dysregulation. Second item is insulin resistance. Insulin resistance, common in obesity, may contribute to psoriasis by affecting keratinocyte proliferation and inflammation. The third item is adipokines. Adipose tissue secretes adipokines, which can affect immune responses and skin inflammation in psoriasis. Another item is gut microbiota. Obesity-related changes in gut microbiota composition can affect systemic inflammation and exacerbate psoriasis through microbiota skin-axis interactions. Also, we have genetic factors. There is a shared genetic susceptibility between psoriasis and obesity that contribute to their co-occurrence. As regard the molecular link between psoriasis and obesity, we have adipokines. Adipokines can be broadly divided into two primary categories: those classified as metabolically unfavorable, such as resistin, and those categorized as metabolically advantageous, such as leptin and adiponectin. Leptin, an adipokine involved in appetite regulation, inflammation, and atherogenesis, is elevated in obese individuals. Leptin resistance, where high levels of leptin fail to effectively regulate appetite, can lead to excessive eating and consequent weight gain, worsening insulin resistance. Multiple studies involving psoriasis patients have documented elevated levels of leptin, which correlate with both obesity and the severity of the disease. An increased production of T helper 1 and interleukin 17A cytokines, which play a central role in the pathogenesis of psoriasis, was associated with leptin. Adiponectin is another important adipokine. Adiponectin, an anti-inflammatory adipokine, showed a reduced level in psoriasis patients who also had overweight or obesity, diabetes, or metabolic syndrome. Adiponectin plays an important role in preventing insulin resistance by suppressing inflammation. It has been observed to decrease the synthesis of tumor necrosis factor alpha and interleukin 6 while simultaneously promoting the production of anti-inflammatory agents like interleukin 10. Studies have shown a negative correlation between adiponectin and tumor necrosis factor alpha. Tumor necrosis factor alpha is known to suppress the production of adiponectin, which helps to explain the decreased levels of adiponectin observed in individuals with psoriasis compared to those without the condition. Resistin is another adipokine known for its pro-inflammatory and atherogenic properties. Unlike leptin and adiponectin, resistin can also be produced by keratinocytes. Research has demonstrated that resistin correlates with the severity of psoriasis and can stimulate the production of tumor necrosis factor alpha and interleukin 6, both of which play roles in the pathogenesis of psoriasis. There was a link between body mass index and serum levels of resistance in some researches, and some also have found a positive correlation between resistant levels and waist circumference. Adipose tissue also directly secretes interleukin 1 beta, interleukin 6, and tumor necrosis factor alpha, and monocyte chemoattractant protein 1. It's important to recognize that these pro-inflammatory cytokines can both stimulate the production of harmful adipokines and suppress the production of favorable adipokines. Obesity can decrease the effectiveness of various psoriasis treatments. Topical treatments may have reduced penetration in individuals with excess adipose tissue. Systemic treatments, such as biologics, may also have altered pharmacokinetics in obese patients, potentially necessitating dose adjustments for optimal results. Insulin resistance, common in obesity, has been associated with the development and severity of psoriasis. It can impede the effectiveness of certain treatments by disrupting cellular processes involved in skin regeneration and immune regulation. Effects of obesity on inflammation and metabolism can impede psoriasis remission. Persistent low-grade inflammation and metabolic disturbances may weaken the effectiveness of treatments designed to suppress immune responses or modulate inflammatory pathways. Achieving long-term remission may necessitate addressing obesity-related factors in conjunction with traditional psoriasis therapies. The treatment response and occurrence of adverse events in psoriasis can be affected by overweight or obesity. This may be attributed to the presence of non-alcoholic fatty liver disease in these patients and the fact that several medications have dosing based on weight. So let's start by methotrexate. In psoriasis patients treated with methotrexate, obesity has been identified as a higher risk factor for hepatotoxicity compared to alcohol consumption, viral hepatitis, and the cumulative dose of methotrexate. This risk is particularly heightened when additional factors such as smoking or diabetes mellitus are present. Recommendations suggest more frequent liver transaminase examinations and consideration of liver biopsies in obese psoriasis patients, especially at cumulative methotrexate doses below 1.5 grams. As regards cyclosporin, obese individuals are more likely to have elevated serum levels, increasing their risk of nephrotoxicity compared to those of normal weight. Studies have shown that modest weight reduction, equivalent to 5 to 10% of body weight, led to improved treatment responses when lower doses of cyclosporin were used in obese patients with moderate to severe psoriasis. Another important psoriatic treatment is acitretin. The impact of acitretin on lipid and glucose metabolism was investigated in a group of 10 psoriasis patients over a span of 1 and 3 months of treatment. The alterations noted in glucose tolerance and lipid metabolism were temporary and showed no correlation with the changes in body mass index or the levels of tumor necrosis factor alpha or obesity-related hormones like resistin and adiponectin. As regard biologics in psoriasis, a multi-center retrospective study involving 504 psoriatic patients treated with biologics found that individuals with body mass index less than 30 were more likely to achieve nearly complete resolution compared to obese subjects. Regarding body mass index, the long-term efficacy of secukinumab and ustekinumab showed a drop. In contrast, adalimumab and etanercept were not negatively affected. Semaglutide is an approved treatment for type 2 diabetes mellitus, acting as a long-acting agonist of the glucagon-like peptide 1 receptor. It is also highly effective in treating obesity by enhancing feelings of fullness and reducing appetite. Glucagon like peptide 1 agonists have been shown to improve clinical psoriasis in a small number of patients with type 2 diabetes mellitus and obesity, correlating with a reduction in interleukin-17 levels and gamma delta T cell numbers. Now let's talk about the microbiome in psoriasis. The microbiome and its link to psoriasis is a growing topic of medical research. The microbiome, comprising bacteria, viruses, and fungi residing in and on the human body, significantly affects health. Recent studies have identified differences in the skin microbiome composition of psoriasis patients compared to healthy individuals. Certain bacterial species are either more or less abundant in these patients. This microbial imbalance can affect the immune system and inflammation, contributing to the development and progression of psoriasis. The human microbiome, comprising, as I already said, all microorganisms living in and on the body, is affected by nutrition and obesity. Mechanisms such as whole bacterium translocation, the accumulation of bacterial metabolites, short-chain fatty acids, and altered tryptophan metabolism link microbiome imbalances to autoimmune conditions. Infections with group A beta hemolytic streptococci, Staphylococcus aureus, and fungi are well-known triggers and aggravating factors for psoriasis. The gut microbiome affects interleukin-17 production, which may contribute to psoriasis pathogenesis. T helper 17 cells and their cytokines play a key role in the progression of post-psoriasis and the mechanisms underlying obesity. This indicates that probiotics, which are effective in managing obesity, might also serve as a promising treatment for psoriasis. Microbiomic data could offer precise molecular signatures for diagnosing psoriasis through DNA sequencing and downstream analysis protocols. In particular, psoriasis is characterized by an overrepresentation of Escherichia coli and an underrepresentation of Coprococcus genus. The gut microbiome of obese individuals shows lower diversity compared to that of non-obese individuals. This reduced diversity is characterized by a decrease in gram-negative bacteria, particularly those in the Bacteroidetes group, and an increase in gram-positive Firmicutes bacteria, which are also prevalent in psoriasis patients. However, research is still in its early stages, and the complex interactions between the microbiome and psoriasis need further exploration. In the future, personalized treatments that consider an individual's microbiome could offer a promising approach to managing psoriasis. So, in summary, a strong link exists between obesity and psoriasis. It is reasonable to suggest that obesity may heighten the likelihood of developing psoriasis, and conversely, psoriasis may contribute to obesity. The pathophysiology of both conditions is interconnected via common inflammatory pathways and immune system dysregulation. Understanding these complex relationships is essential for providing holistic care to individuals having both conditions. Applying a multidisciplinary strategy that tackles the underlying mechanisms of both psoriasis and obesity is vital for enhancing patient outcomes and overall quality of life. Lastly, I hope that you have enjoyed this important video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.